it's Gabrielle, and this video is a sample from Studyclix Boost, our new rapid revision tool. Go to studyclix.ie/boost to find out more.
Osmosis is defined as the movement of water molecules from a region of high water concentration to a region of low water concentration across a selectively permeable membrane. In this Leaving Cert Biology experiment, we will use Visking Tubing to act as an artificial selectively permeable membrane. It is used to mimic how a cell membrane works. To prepare the Visking tubing, it must be softened in water for about 5 minutes and then opened. After knotting one end, using a syringe, we add an 80% sugar solution, such as sucrose, to the tubing. This has a high solute concentration and therefore a low water concentration. We knot the other end and find the mass of the Visking tubing using a mass balance. A quick note here, make sure to dry the outside of the tube beforehand to make sure the mass is accurate. Our control for this experiment is Visking tubing filled with distilled water. Again, we must find the mass of this tube also. We place both of these tubes into separate beakers with distilled water and leave them for about 30 minutes. Be sure to keep the ends of the Visking tubing out of the distilled water in case some of the solution leaks out. You can prop them up using glass rods or pencils. We then find the mass of both tubes again after 30 minutes. You can see the expected results here are shown in the table. The Visking tubing with the sucrose solution has increased in mass and turgidity due to water moving from the beaker into the tubing by osmosis. So we know that osmosis has taken place as the mass of the Visking tubing is increased and the tube is more turgid. You can also say that it feels fuller. In the control, you can see that there is no change in mass or turgidity. This is because we had the same water concentration either side of the Visking tubing, so there was no loss or gain in water. So what do you need to know for the exam from this experiment? Well, explain what Visking tubing is and why we use it for this experiment. Outline how we set up the experiment, making sure to include the control also. And describe how we know that osmosis has taken place in this experiment.